Hi everyone, myself Shana Shaji, second MSc Biology, St. Mary's College, Thrissur. Today we discussing about the topics endoplasmic reticulum and Golgi bodies. Now we discussing about endoplasmic reticulum. Morphology or structure. Endoplasmic reticulum is a vast network of membrane-bound, branching and interconnecting tubules, vesicles and flattened sacs. It is irregularly distributed in the cytoplasmic matrix. The term endoplasmic reticulum was coined by Porter in 1953. There are two kinds of endoplasmic reticulum: granular or rough endoplasmic reticulum and agranular or smooth endoplasmic reticulum. This is the picture of endoplasmic reticulum. Here we can see that smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, cisterna, and the cisternal space. Then move to the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum, which means the ribosomes is present, composed of network of flattened sacs, that is cisterna. It is a location for the synthesis of the protein constituents of endoplasmic reticulum. It may develop from nuclear membrane, abundantly found in exocrine pancreatic cells and the antibody secreting plasmid cells, and finally pores are present. Then move to smooth endoplasmic reticulum. Here the ribosome is absent and it is mainly composed of tubules. It is a location for the synthesis of non-protein substances. It may develop from rough endoplasmic reticulum, abundantly found in liver and testicular cells, that is synthesis of steroids, and pores are absent. This is a picture of smooth endoplasmic reticulum and the rough endoplasmic reticulum. Here we can see that the ribosome is absent in smooth endoplasmic reticulum and the ribosome is present in rough endoplasmic reticulum. And the endoplasmic reticulum exists in three morphological different forms, namely lamellae, vesicles, tubules. Lamellae are unbranched and flattened sacs, that is arranged in the parallel stacks, and their internal lumen is called cisterna. Vesicles. Vesicles are closed oval or rounded sacs. They are found in large number in liver cells and pancreatic cells. Tubules. There are the unbranched and interconnecting tubules, which is abundant in cells which are active in the synthesis of steroids. The tubules and vesicles are mostly granular, whereas lamella are agranular. The lamellar and tubular units may merge together to form a continuous network. The cells are homogenized and get fragmented. These fragments soon get reorganized, forming small vesicles called microsomes. This is a picture of lamellae, vesicles, and tubules. The chemical makeup and the molecular organization of endoplasmic reticulum. Here, the membranes are endoplasmic reticulum have lipoprotein composition and the fluid mosaic organization. They are formed mostly of phospholipids, cholesterol, intrinsic and extrinsic proteins. Phospholipids forms a bilayer. Cholesterol, which binds with phospholipid bilayer to form a stable complex. Intrinsic proteins partially or completely penetrate the lipid bilayer, and extrinsic proteins remain entirely outside the lipid bilayer. Intrinsic proteins are mostly structural and the extrinsic proteins are mostly enzymatic. There are two characteristic transmembrane structural glycoprotein in the rough endoplasmic reticulum. There are riboflavin 1 and 2, but which are absent in smooth endoplasmic reticulum. Riboflavin 1 and 2 serve as a site for ribosomes. Then this is the sarcoplasmic reticulum. It is a membrane-bound structure found within muscle cells which are similar to the smooth endoplasmic reticulum in other cells. Sarcoplasmic reticulum is an extensive network of specialized smooth endoplasmic reticulum found in striated muscles, which is mainly concerned with the storage of calcium 2 plus ions. Calcium 2 plus ions are stored in the endoplasmic reticulum with the help of calcium binding proteins. Then finally, functions. The endoplasmic reticulum provides a structural framework and the supplementary mechanical support to the colloidal cytoplasmic matrix. Collection, storage, chemical processing, and the intracellular transport of secretory proteins, regulation of ionic gradients, membrane potential, and the intracellular pH, synthesis of phospholipids, lipoprotein, glycerin, sterol, steroid hormones, etc. Metabolism of vitamin A and synthesis of visual pigments in retinal cells. Transport of RNAs and the ribonucleoproteins from nucleus to cytoplasm. These are the main function of endoplasmic reticulum. Then move to the Golgi bodies. Golgi bodies are aggregation of membrane-bound spaces particularly abundant in secretory cells. The Golgi bodies were first discovered and described by Camillo Golgi in 1898. Plant Golgi bodies are often described as dictyosomes. Golgi bodies have no attached ribosomes. Their membrane appear to be surrounded by a small area called the zone of exclusion. The morphology or structure of Golgi bodies. Golgi bodies are essentially membrane-bound spaces. There are three kinds, namely lamellae, vesicles, and vacuoles. We know that lamellae form the core units of Golgi bodies. Their internal lumen of lamella is called cisterna. Lamella are flattened sacs with dilated ends. They are arranged in parallel stacks or piles. Are called Golgi zones. Vesicles are a group of small membrane-bound spaces. Vacuoles are large and scattered membrane-bound sacs. This is a picture of Golgi bodies. Here we can see that incoming transport vesicle, lumen, newly formed vesicle, tubules, trans or maturing phase, and the cis or forming phase, cisterna, forming Golgi and vacuole, coated vesicle, secreted vesicle. Then each lamella has a polarized orientation. It has a convex outer face known as forming phase, also known as entry phase or cis phase. Concave inner phase known as maturing phase, also known as exit phase or trans phase. Each phase is closely associated with a network of tubules forming cis Golgi network and trans Golgi network. Forming phase. It receives vesicles from endoplasmic reticulum and it is oriented towards rough endoplasmic reticulum or nuclear envelope. Maturing phase, which defined as it gives out a large number of secretory vessels, it is oriented towards the cell surface. This picture shows the secretory role of Golgi bodies. A lamellar sac contains three major kinds of sacs with different functions. They are cis sac, trans sac, and medial sac. Cis sac, which is located towards the outer or cis face of the sac. The function of the cis sac mainly concerned with the phosphorylation of mannose molecules of the glycoproteins that are destined for lysosomes. Trans sac, which is located towards the inner or trans sac, and it is involved in the addition of galactose and sialic acid to glycoproteins. And finally, medial sac, which is located in the middle of the sac and function is involved in the removal of mannose molecules and the addition of acetyl glucosamine to glycoproteins that are destined for the exocytosis through plasma membrane. The chemical composition, which means the Golgi bodies are chemically unstable. Their chemical makeup may change from time to time. The membrane have lipoprotein composition, laminar structure, and fluid mosaic organization. Golgi bodies are formed mainly of proteins and phospholipids in equal amounts, which are the variety of enzymes. The enzymes found in high concentration include thiamine, pyrophosphatases, and glycosylation transferases. Finally, the function of Golgi bodies. Golgi bodies are important chemical factories or secretory centers of the cell. It is a removal and recycling of excess membranes and secretory granules. formation, packaging, transport of cell wall polysaccharides and the petting components for cell wall formation plant cells, secretion of mucus, milk protein, thyroxine compounds, formation of ectosome in developing sperms, formation of microbodies and primary lysosomes, activation of mitochondria. Thank you.